This is my presentation for plant anatomy and growth, which is chapter 20. So before I get into the actual chapter, I want to go over some background information, which is going to be about the pyramid scheme that makes up organisms. So as we can see in the PowerPoint, we first start out with cells, which are made of organelles, and cells come together to make different types of specialized tissue, like we see here, and then tissues come together to make specialized organs. An example can be a leaf, and then different organs come together to make an organ system, which can be known as the flower of a plant, and then all those organ systems together make up one organism. And so here is the organism. I have a bean plant, which is what I'm going to be doing my experiment over. And so a quick note I want to make is that when a plant first begins to grow, the first cells are called meristem cells. So the first part of chapter 20 talks about plant cells and tissues. And so the meristem cells organize themselves into meristem tissue that allows the plant to grow throughout its life, and it's usually present at the top and the bottom of a plant. And these ends, the top and bottom, are called apical meristems, which differentiate themselves into three types of tissues. The first type is called epidermal tissue, which forms the protective covering of a plant. The second is called ground tissue, and it fills the inside of a plant and helps carry out other functions. And the last is called vascular tissue, which transports water and nutrients inside the plant and provides structural support. Next, we're going to take a closer look and understanding of epidermal tissue. So the whole plant is covered by this tissue, and it acts as a barrier, like our skin, which is also called epidermal skin. So the parts that are exposed to air have a waxy cuticle that minimize water loss, protect against bacteria, protects against other organisms that cause disease. Epidermal cells can be further specialized into things like root hairs that are long, thin, and increase the surface area for better water absorption and minerals. This is usually seen in the roots. In leaves, there are stomata, which are small openings, and they are surrounded by two guard cells that serve as a site for gas exchanges and anywhere that water loss happens. Epidermal cells can also be trichomes, which makes stems feel prickly or sticky, which scare off any harmful insects or other predator animals. A closer understanding of ground tissue. So ground tissue makes up the inside of leaves, stems, and roots, and they contain three types of cells. The first is called the parenchyma, which is the least specialized, and it's found in all organs of plants, can contain chloroplasts and carry out photosynthesis, and can have colorless organelles that store photosynthesis products. Collenchyma, which is like the first type, except they have irregular corners, and they have thicker protective cell walls, and they form bundles under the first layer of skin of a plant, which allow flexibility in young plants. Sclerenchyma have a second thick cell wall, and they have lignin, which makes a plant cell hard. So most of these cells are non-living because their only purpose is to provide support to mature plants. A closer understanding of vascular tissue. Vascular tissue can be stretched throughout the root, from the root to the stems, and from the stems to the leaves, and vice versa. In the root, vascular tissue is in the central cylinder. In the stem, vascular tissue is found in bundles, and in the leaves, vascular tissue can be found in leaf veins. So the tissue can be further broken down into the types of xylem and phloem. Xylem is in charge of transporting water and minerals from the root to the leaves, and phloem is in charge of transporting sugar or sucrose and other organic compounds from hormonal leaves to the roots. Xylem is further broken down into conducting cells like vessel elements, which are larger, they're perforated, and they're arranged like a pipeline for transportation. And they can also be broken down into tracheids, which are like pits that allow water to pass. So, first we're going to take a look at the first peer-reviewed article. Like previously we mentioned when talking about ground tissue, we know that throughout a plant's growth, plants need movement. One thing that I find interesting is how fast plants can move. And one example can be plants that are nyctinastic. Nyctinastic plants open and close their leaves with a circadian rhythm. Motor cells play an important role in this. So motor cells are a type of plant cell that act like a hinge at joints to enable the movement of plant parts, such as closing and opening of leaflets in response to light, intensity, or rapid closure of leaf in a carnivorous plant. So as we can see, the first picture, the carnivorous plant is closing its leaves very fast to capture its prey, and then the second one, that's a prayer plant, which is responding to the light dimming. So whenever light dims, it closes its leaves. So from that same article, I was able to get some information of the place where the leaves close. So the site is called a turgor. So whenever they swell, the leaves open, and whenever they shrink, the leaves will close with them. So the first two pictures shows a real plant and where the leaves have closed in on themselves, and the bottom picture shows an animated version of how the turgor either swells or shrinks, which affects the motion of leaves. So this is connecting with the peer-reviewed article, which is around bean plants. The research in this article was about monitoring bean growth and things that make beans grow stronger, faster, and more efficiently. More specifically, the article used chemical treatments rather than physical treatments. They use PGRs, or plant growth regulators, to modify growth and development. A quote from the article is that these physiological and biological substances, even at very low concentrations, trigger and coordinate many basic and physiological processes in the plant life cycle, such as cell division, cell enlargement, and differentiation, organogenesis, seed dormancy and germination, leaves senescence and abscission, as well as defend against abiotic and biotic stresses. I will perform an experiment with different physical treatments to show how bean growth can be affected. So, a breakdown of my experiment with bean plants. The point of this experiment is to show how different physical environments affect the growth of different pinto bean plants. There will be a total of 5 different environments. The first will be a bean placed into soil and water with no direct sunlight. The second will be a bean placed in water, sun, and with a moist napkin inside of a Ziploc bag. The third will be a bean in a Ziploc bag with only water and sun. The fourth is going to be a bean with pebbles, water, and direct sun. And the last one is just going to be soil, water, and sun. My hypothesis is that all beans will grow relatively the same, except for the bean in the pebbles and the one submerged in water. These two. Because soil and humidity should be enough for them to sprout. I say that the bean in the pebbles will not sprout because there isn't enough humidity and the bean in water will not sprout because the beans are seeds that are not supposed to be soaked, and that bean will be submerged. So here we have the first day of the experiment. Here we have the bean with water, sun, and a napkin taped to the window, and the bean with water and sun taped to the window. And here we have the soil, water, and sun, and the pebbles and sun with direct sunlight. And lastly, it's the one with only soil and water in a dark room. On day 2, on April 16th, the only noticeable change is that on the first bag, you can see humidity that is forming because of the wet napkin, which will be a great at sparking growth in the seed. Humidity is important in the transpiration process, which is how plants get water vapor to feed the plant. And in the second picture, you can see how there's no humidity, which will probably lead to the plant not being able to grow. By day 4, the seed with the napkin had sprouted a root, and the seed with water still had zero humidity, as we can see right here. And the rest of the beans looked the same as the first day, so I didn't take a picture. And on this picture, I do want to note that the root growing downwards is an example of geotropism, which is where the root goes down, and then soon we'll see this part of the bean grow upwards. By day 6, the seed with the napkin has even greater root growth. We can see the main root and lateral roots, and we can start to see the actual top of the green plant start to green. The second bag has the same root growth right here, but this one does not because it had so much water around it. And again, the other beans had no growth, so I did not take any pictures. On day 7th, April 21st, the bean with the napkin had great growth. As you can see the roots, you can also see hairs right there. And then the bean that was submerged in water, it had a small root, but you can tell that it fell off. And the bean has been starting to explode because of too much soaking in the water. And the bean that was in soil and water and indirect sunlight had finally started to show some signs of growth. And lastly, the beans in the pebbles and water and sun, and the soil, water, and sun didn't really have any growth, but in this bean you could start to see the division between the cotyledons. By day 9, I took the seed with the napkin out of the bag and placed it into soil so that it can actually produce beans in the future. And in the picture right here, you can see the elongation of the stem. You can see the 2 separate cotyledons, and in between the cotyledons you can see the first true leaves. And then in these beans, you can see how this one is starting to bubble because it's starting to rot and explode. And the other beans had no noticeable growth. For days 9 through 11, which was April 23rd through the 25th, I decided to include a small series of pictures that show how fast the cotyledons opened up and the true leaves began to open up. So here we can see that in the same day, in just 4 hours, the true leaves were able to open up, and then by the next day, there was elongation of the stem. Here are the true leaves, and the cotyledons have completely opened up, and then by the next day, there was even greater growth here, and the true leaves were starting to open up. On day 13, the bean with only soil and water had finally sprouted. The rest of the beans had no significant growth. This is the bean that was originally with the napkin. You can see how the cotyledons right here had shriveled up, and here are the mature true leaves. And then this is the bean. This bean and this bean are the same, but this is by day 17. The bean has started to experience phototropism because it needed and it wanted light. So my conclusions for this experiment. So throughout this experiment and observation, I was able to prove my original hypothesis that all beans will grow relatively the same, except for the bean in the pebbles and the one submerged in water, because of soil and humidity, were wrong. So I was able to see that just because some seeds had nutrients from soil, they mostly needed humidity, like the bean with the napkin, which would end up being the healthiest and the biggest growing plant. It is also important to know that maybe some beans didn't sprout because they were too old or they were just bad beans. I was even able to see one plant experience phototropism, which is where the growth of a plant starts to go toward light, which is what you could see here. It was starting to go towards the window instead of straight up like the one that was by direct sunlight. And that was all that I was able to get for my experiment. So I hope you enjoyed. And this is my works cited.